Master's student in my lab. Comes from the northland of Minnesota and actually went further north for his undergrad at University of North Dakota, not North Dakota State. And then he has been here for the last 2+ years working in the lab, and then he's working on SDS like a lot of other people in the lab. And so sit back and relax, enjoy it. Dan, how are you?
All right. Hello, everybody, and welcome. Thank you for coming. My name is Daniel Sjarpe, and today I will be talking to you about the effects of seed treatment and soybean varieties on root rot caused by Fusarium virguliforme. A brief outline is, what is SDS? Followed by, why does it matter? Then we will talk about the disease cycle of SDS. Then we'll get into my personal studies, first the greenhouse study, then the field study, and then we'll get into our conclusions. So, what is SDS? SDS is sudden death syndrome of soybeans. It is caused by Fusarium virguliforme, here on called FV, which is an ascomycete fungi that is soil-borne. The optimal disease conditions are cool and wet conditions at planting, and then heavy rainfalls during the reproductive stages to make the foliar symptoms occur. So, distribution of SDS. It was originally found in Arkansas in 1971, and has since spread to nearly every soybean producing state. It was first found in Iowa in 1993, and interestingly enough, it was also first found in Canada that same year. Now, why should we care? You can have yield losses of up to 100% on a field basis. And SDS is the second-ranked disease for yield across the past 5 years. And between 2010 and 2014, there was over 200 million bushels lost due to SDS. Please keep that number in mind for the end presentation. And in 2014, over 63 million bushels were lost that year alone. On to the disease cycle. We will be using this same picture for the next several slides, so if you cannot see the writing, don't worry, I'll explain it a little better. So, inoculum survives on soybean and corn residue, and it also overwinters in the soil. It overwinters as cometospores and macroconidia, as seen in the top picture. Now, this bottom picture actually shows FV growing on an auger plate, and you get this blue sporulation. It is important to note that SDS is made up of 2 phases, both a root rot phase and a foliar symptom phase. So for the root rot phase of the disease, it infects soybean roots very early on in the season. And this picture shows a root much later in the season, actually towards the very end. But why I put it here is that you can see this blue fungal growth that's followed by the arrow. Another few things of note for this particular root, so you can see just how rotten it is. This is not the color a root should be. This is all darkened. And the third thing to take away from this picture is that there's almost no lateral roots whatsoever. You just see a few here and here. So, once the pathogen attacks the roots, it colonizes only in the root tissue. It does not move higher than that. So you might be asking yourself, how do you get those foliar tissues? The pathogen itself produces a toxin that needs mid-season rain later on to move up into the foliar symptoms. Now, for the toxin to move, this means that the pathogen must colonize the xylem so that the pathogen can actually, so that the toxin itself can actually move up and cause those foliar symptoms. Here is a picture of a characteristic foliar symptoms, which you get the intervenal chlorosis and necrosis, which typically starts around the R2 phase. And symptoms first appear in the upper canopy and move down from there. Now, once again, this is a characteristics of sudden death syndrome, but if it gets bad enough, you can actually get complete premature defoliation, along with pod abortion, and the seeds that are left, and the pods that are left behind will have smaller seeds in them. All of these lead to yield loss. Now, one interesting thing about SDS is that the leaves themselves fall off, leaving the petioles behind. And this picture is actually taken from just outside of Ames here, and it shows a field with the high SDS pressure. Now, you can see two green patches here and here that look like they have relatively no disease. You can see through the ravine, right through here, that it has high SDS pressure, and this picture is actually a great way to show that SDS is FE is a soil-borne pathogen, and that needs that early season rain, which low spots in fields naturally have higher water amounts. There are many management options available for SDS. However, none of these are a magic bullet. That's why there has been so many different management options to help try and reduce these. These management options include planting a resistant variety, seed treatments, SCN management using resistant varieties, and then there are also several cultural practices available, including crop rotation, tillage practices, and changing the planting day. Now, this table actually shows all of those management practices from the last slide, but it's actually broken up into the root rot phase and the foliar phase of SDS. So, for resistant varieties, there's been no effects reported on the root rot phase of SDS. However, for foliar symptoms, there are many varieties available that reduce foliar symptoms. For seed treatments, Fusarium has been shown to both reduce root rot and the foliar symptoms. There are several other seed treatments that are labeled against SDS with varying levels of results. For SCN management, I could not find anything in the literature for specifically controlling the root rot phase of SDS. However, SCN presence does increase foliar symptoms. On to crop rotation. Once again, I did not find anything in the literature about crop rotation specifically reducing the amount of root rot. But when rotating away from the corn-soybean rotation, you can actually reduce foliar symptoms. On to tillage. No effect was found for tilling on the root rot phase of SDS, but tillage can help reduce foliar symptoms in compacted soil. However, these results vary depending on which paper you have read. For adjusting planting date, if you change your planting date to later, it can reduce root rot. And it has also been shown that early planting can increase foliar symptoms. But these results, once again, vary depending on which paper you read. Now, it is important to note that there is, on this table is that there are many studies that have been done on the foliar symptoms, but not nearly as many has been done on the root rot phase, as there were 2 blank that I couldn't find in the literature, and also 2 of the 4 that did have something show that no effects were reported on the root rot phase. Which brings me to the goals of my research. The goals of my research are to identify a quick greenhouse screen test for screening seed treatments. The next goal is to determine the effect of the root rot phase of SDS on yield. And the third goal is to understand how Fusarium seed treatment and resistant varieties affect both phases, so both the root rot and the foliar symptoms of SDS. On to my studies. So this is the same between both the greenhouse studies and the field studies. 3 seed treatments were used. The base seed treatment, which is Evergold Energy, Allegiance, plus Pancho Votivo, which these are insecticides, nematicides, and fungicides. And it will, from here on out, be referred to as base. The next seed treatment is base plus a low rate of Olivo, which is Fusarium. And the third one is the base plus a high rate of Fusarium. Now, it's important to note that when we do run just the base, this has been studied and there has been no effects on SDS itself. So it is the Olivo for the later slides. There are also 3 varieties chosen, but they varied by location. But they have the same theme in common. They had a susceptible, a moderately resistant, and a resistant variety. Now, on to the greenhouse study, which once again, the goal is to identify a greenhouse protocol to evaluate seed treatments. 2 technique study. We used those same 3 seed treatments as previously stated, so the base plus high Fusarium, base plus low Fusarium. And had 3 soybean varieties, the susceptible, moderately resistant, and resistant. And 2 to 3 inoculum levels were used. Both used inoculated and non-inoculated. The inoculated was FE-infested white sorghum, And the layer technique included a third inoculation level, which was the mock inoculated, which was the white sorghum that was autoclaved twice to mimic the inoculum. The setup for both of these was 6 reps were completed per run, and 2 runs were completed per experiment. They were completed in a randomized complete block design. Plants were allowed to grow for 30 days, and from there we rated SDS foliar symptoms and the root rot. For the soil incorporated inoculum, the ratio we used was 30 parts soil to 1 part inoculum. So what we did is we added 30 parts of the soil, and we added in that 1 part of the inoculum, and we mixed until homogeneous. Now 5 plants per pot were planted, and then weeded to 3 so that we had consistency throughout. Now, all of these graphs for the greenhouse studies are very similar, so I'll go in depth and explain this. For the base seed treatment is this dark purple on the left, the high Fusarium is the blue, the low Fusarium seed treatment is the orange, the susceptible is this pink, I apologize for the colors, this is pink, this moderately resistant is right here, and that is green, and then this light purple is the resistant variety. Now, there was very little foliar or root rot for either the non-inoculated or the mock inoculated. So the only thing shown in the graphs are the inoculated. Another thing to note throughout is that for each seed treatment, this is the average across all 3 varieties. And the same for the variety. Each variety had all 3 seed treatments on it, and that shows the average. If there was no significant difference at a p-level of 0.05, we included the p-value in parentheses above the group of 3. So now onto this particular graph. This graph shows the mean foliar disease severity based in percent. Unfortunately, neither the 3 seed treatments or the 3 varieties were significant. However, there was still between 20 and 30% foliar severity. Once again, this slide follows the same color scheme, with the dark purple being the base, blue being high Fusarium, orange being low Fusarium, this pinkish being susceptible, this green being moderately resistant, and this light purple being resistant. For this graph, you can see that the base seed treatment had significantly higher root rot than either the high rate or the low rate of Fusarium. The Fusarium actually had a 20 to 25% reduction in root rot compared to the base. Now, onto the layer technique. So why is it called the layer technique? These were grown in 7-inch containers, and what was done was we filled the first 5 inches with soil, and then we added a layer of the different inoculum levels. From there, we added 1 inch more of soil. We placed the seed on top of that, and then we filled the cone until full. And from there, when we watered it, they shrink down a little, as you can see in these pictures. Now, the reason for this layer of the inoculum 1 inch below the seed is that it forces that seed at a very early stage to grow its roots through that inoculum. Another important note for the layer technique compared to the soil incorporated inoculum is that it was done in a water bath. The water bath held from 17 to 19 degrees Celsius, which was the closest to the optimum temperature for the root rot phase of SDS that we could get in the greenhouse. Once again, this graph is similar from the last ones, with the dark purple being the base, blue high Fusarium, orange low Fusarium. This pink is susceptible, green is moderately resistant, and this light is resistant. And once again, only the inoculated is shown due to the very low foliar and root rot of the other inoculum types. So, what this particular graph shows is the mean foliar disease severity. And you can see that the varieties themselves had a significant difference between the moderately resistant variety in group A and the resistant variety in group B, while the seed treatment was not significant. When we go to the mean root rot severity, once again, base dark purple, blue high Fusarium, orange low Fusarium, pink susceptible, green, moderately resistant, and light purple resistant. When you get to the mean root rot severity, you can see that there was a significant reduction in root rot between the base seed treatment in group A and either of the rates of Fusarium. There was actually roughly a 20% reduction in root rot with Fusarium compared to the base. Another important thing to note is that the layer technique had a much higher root rot severity than the soy incorporated inoculum. It was actually roughly twice as much root rot severity. For our greenhouse results, we found that neither technique consistently differentiated between the treatments for the foliar symptoms. However, both techniques showed an approximately 20% reduction in root rot severity between the base seed treatment and both rates of the Fusarium. Further work is needed to ID a reliable greenhouse screen for seed treatments, but our lab is currently working on that with an aeroponics system that is being set up in the greenhouse. Now, on to our field study. Field studies were completed in 2015 and 2016 at 9 different locations. There were 5 locations in 2015 and 4 locations in 2016. These locations included 2 locations in Iowa, and then it also included Minnesota and Ontario, Canada. The same 3 seed treatments as before were included, so the base plus high levo, base plus low levo. And then 3 soybean varieties which varied by location, but still had the susceptible, moderately resistant, and resistant throughout. Foliar symptoms were collected on a weekly basis as soon as they showed up. We collected root rot at several different stages, and at each location that we collected root rot, we also collected root weight. We ran qPCR on those roots. And we did all of this to see how all the different variables affect yield. On to the field layout. The fields are laid out in a randomized complete block design in a strip plot arrangement, with the vertical strips being the seed treatment and the horizontal strips being the variety. And there were 4 reps per location. Now this picture actually shows our location in Roland, Iowa in 2015. And what I find very interesting is you can see all of these different 4-row strips, because that's how wide the planter is, in 4 rows, that are very, very heavily infested with the SDS, which shows just how much SDS was out in the field that year at that location. For foliar symptoms, we first found the disease incidence, which is the percent of the symptomatic plants per plot, and then we found disease severity from there on those infected plants on a 1 to 9 scale. From there, we can calculate FDX, which is disease incidence times disease severity divided by 9. For our root rot ratings, we collected 10 roots at multiple timings throughout the year. And what we did is we brought them back to the greenhouse, we washed them, and then we rated them using a visual 0 to 100 scale. What this picture shows is representative of the scale we used, and this picture shows anywhere from 10% root rot all the way up to 100 in 10% increments. After the root ratings were completed, we allowed the roots to air dry in the greenhouse for about 1 week, and then we weighed them using a digital scale. After weighing the roots, DNA was extracted from the uppermost 5 centimeters of the taproot, and then they were processed using the DNeasy plant mini kit from Kyoto. From there, qPCR was run with a specific assay to FV. For yield, we harvested the middle 2 rows, grain moisture was recorded, and then it was adjusted to 13% moisture to standardize yield across all the different locations. Yield loss, as described how we found it on the last slide. It is important to note that this is actually the order of this. I know it might not make sense with the years being mixed up, but the order of this actually goes from the lowest FDX all the way to the highest FDX. This ranged anywhere from 0 in Highgate, Ontario, all the way up to over 50% root rot in Rosemount, Minnesota. Now for the locations, HGON is Highgate, Ontario, Muscatine, Iowa, Rodney, Ontario, Rosemount, Minnesota, and Roland, Iowa. So, after we ran the linear regression on all of those different variables, what it gave us was these 3 variables were the most significant, were the best explanatory variables for the yield loss. And those 3 variables are root rot severity at R5, FDX at R6, and dry root weight at R5. Now the numbers in the colored boxes actually show the R squared number. And what that means is this number is the percent that explains this. So what that means is for Highgate, Ontario, root rot severity accounted for 23.2% of that yield loss. So 23.2% is roughly 25% for easier math. So what that means is that the root rot severity accounted for 1/4 of this, or 2.2% of the total yield loss at that location. Now, as you can see here, 5 of the 9 locations had root rot severity at R5 being the significant, with the significance ranging, with the R squared ranging anywhere from 4.6 all the way up to the 23.2. While in FDX, 6 of the 9 locations had FDX be significant, ranging anywhere, the R squared ranging anywhere from 10% all the way up to 88.5. Now, as previously mentioned on this is in order of FDX, and it's not on here, but both of these boxes, actually there was no FDX at those locations. So in actuality, it was 6 of the 7 locations where FDX occurred had FDX being significant. Now for the dry root weight, it was significant at 3 of the 6 locations, but at 2 locations, dry root weight actually increased yield, which is the yellow boxes, but at 1 location it actually reduced yield, which brings me to the next slide. So the summary for that regression is that SDS caused an average of 17.2% of the yield loss, and root rot reduced yield at 5 of the 9 locations. And there was an average of 9.6% of the yield loss from these fields. This means that 13% of SDS yield loss is due to the root rot phase. It was also found that FDX reduced yield at 6 of the 9 locations, with an average of 62% of the yield loss from these fields. So this means that 87% of the SDS yield loss is due to the foliar phase. And then other variables were not reliable in predicting yield, as they either did not make the model or had the results like the dry root weight. So now on to this. So why we ran those 3 variables, excuse me, is that it showed what was most important. So that is what we showed. So we will be showing you the mean root rot severity. So these graphs are all set up very, very similar, so the blue is the base seed treatment, red is high fluparum, and green is low fluparum. Now we only show the Iowa data for the purpose of this presentation. So we have Muscatine, Iowa, and Roland, Iowa. And it's always set up at 2015 on the left, 2016 on the right. So what this particular graph shows is the percent root rot. And as you can see by the groupings, it was significant at all 4 locations. And at all 4 locations, the base seed treatment had significantly higher root rot than either rate of fluparum. It is interesting to note that Muscatine, Iowa in 2015, the high rate of fluparum actually had significantly less root rot than even the low rate of fluparum. Now this reduction on average was between 40 to 50% root rot reduction between the base seed treatment and the different fluparum rates. Now, this graph also shows mean root rot severity, but this graph is broken up by variety. So, on this graph, the blue shows a susceptible variety, red shows a moderately resistant variety, and green shows the resistant variety. Once again, it's set up with only Muscatine and Roland, Iowa. And as you can see, only the Roland, Iowa location was significant. However, the moderately resistant variety was the only one that had significantly less root rot than either the susceptible or the resistant variety. This graph is perfect for stressing that breeders select for SDS resistance based off the foliar phase of SDS and not the root rot phase. This graph shows the mean FDX by seed treatment. So we are back to seed treatment. Once again, blue is base, red is high fluparum, and green is low fluparum. FDX, there was significant difference in FDX at both locations in 2015. Once again, with the base seed treatment having significantly more FDX than either rate of fluparum. Neither location was significant in 2016, and it is also important to note that there was very little FDX in Muscatine, Iowa either year, as shown by the lack of bars. On to the mean FDX by variety. Once again, we're back to variety. So the blue is susceptible, red is moderately resistant, and green is resistant. FDX, there is a significant difference at 3 of the 4 locations. Both locations in 2015 had the susceptible variety being higher than the moderately resistant variety. And in Muscatine, it was higher than both. While at Roland, Iowa, the resistant variety fell into both groups with the A-B distinction. In 2016, in Roland, Iowa, both the susceptible and the moderately resistant variety were in group A, while only the resistant variety actually had less FDX at that location. Now, on to why this is all important. This is the mean yield broken up by seed treatment. Once again, blue is the base, red is high fluparum, and green is low fluparum. So, we'll start with 2015. Both locations had a significant difference in yield, with the high rate of fluparum being significantly higher yield than both the base or the low fluparum seed treatment at Muscatine in 2015, while in Roland, Iowa in 2015, the base had lower yield than either, so both rates of fluparum were higher than the base seed treatment. Neither location was significant in 2016, but it is to note that this difference is actually 11% yield increase with the high fluparum compared to the base seed treatment, and a 5.9% yield increase with the low fluparum compared to the base seed treatment. Now, on to yield by variety. Once again, blue susceptible, red moderately resistant, green resistant. All locations were significant. For Muscatine, Iowa in 2015, both moderately resistant and resistant varieties had significantly higher yield than the susceptible variety. While in Roland, Iowa, only the moderately resistant variety had a significantly higher yield than the other two. In Muscatine, Iowa in 2016, the moderately resistant variety had significantly less yield than either the susceptible or the resistant. And in Roland, Iowa, the resistant variety had significantly higher yield than the other two. Okay, this brings me to my conclusions. So my conclusions were, for the fluparum seed treatment in the greenhouse, there was approximately a 20% reduction of root rot for both the rates compared to the base. But more work is needed in the greenhouse to ID a better seed screen. For the field, there was a 48.7% reduction and a 38% for the high fluparum compared to base, and a 38.5% reduction for the low fluparum compared to the base. There was approximately a 38% reduction of foliar symptoms where they occurred. Now, on to the important part, is that the most important part, is that there was an 11% increase in yield from the high fluparum compared to the base seed treatment, and a 5.9% increase from the low rate of fluparum compared to the base seed treatment over the course of that 5-year span. So what this says is that up to the 26 million, which is 13% of the 200, could be due to the root rot phase of STS alone. With that, I'd like to thank everybody for coming. I'd like to thank my committee, Darren Mueller, Leonor Leandro, Forrest Nutter, and Mark Licht. I'd also like to thank our collaborators, Albert Tenuta up in Canada, and Dean Melvick at the University of Minnesota. I'd also like to thank the Mueller Lab, especially Yuba, for answering all of my many, many questions. I'd also like to thank our funding sources, the United Soybean Board, and Bayer Crop Science. And with that, any questions?
Nice job with that work. I really like your project. I missed a few details of some of the methods. In the greenhouse, your inoculum, was it a sorghum seed carrier?
Yeah, we used the white sorghum.
Okay.
Yep, that was infested with that weed.
In the field, that was natural inoculum?
So some locations were naturally infested, while other locations were inoculated, depending on if the university owned them or not. So we're allowed to do it. Yeah, yeah.
And then remind me how you calculated your disease index. It was the incidence times.
Oh yeah, it's the disease incidence times disease severity. And what you do is you multiply those two and divide by 9, which actually gets it back to that 100 scale.
Right, but the severity rating was on individual plants or for the whole?
Oh, it was for the whole plot.
Okay.
No, severity is that all the plants are infected.
Yeah, so that's why it's a 1.
There's 30% incidence, you just rate those 30% of the plants.
Okay, right. That's what I was getting at. So you're just talking about you rated individual plants.
Yeah.
Okay. And then I don't think you showed any of the qPCR results.
Oh, yeah, I'm sorry, I forgot to mention that. So the reason why we did not include any qPCR data was because when we did the linear regression, we found that the qPCR data was not significant to the yield, so we didn't show that in this experiment.
And did it correspond with the disease index or the root route or anything?
We did a correlation with everything, and it was because we did a lot of qPCR, because we did anywhere from 2 to 4 timings per location with the 9 locations. I don't remember any particular one jumping out right now in my head. I apologize for that.
Essentially, we did like $10,000 worth of qPCR, and it really did nothing.
Yeah.
Yeah.
I imagine if it would have been interesting, it would have been in your presentation.
Yeah, of course, yeah.
So,
I forgot to mention that bullet point.
So that's a sad outcome.
Yeah.
It took all winter each year.
Mark?
Looking forward to seeing. So I have to ask this question related to the disease assessment you had. Dean Malvick and Albert Tenuta and all these people helping you. So you have this 9-part system, right?
9 location system, yeah.
No, I mean, I thought you had a
9-point
qualitative
scale.
rating scale. Did you not?
Yeah.
Oh, yeah, yes.
Yeah, okay, so I don't think you ever showed, and maybe I was dozing at the time, but it was good presentation, I'm told. But, you know, I would have liked to see, or what your visual disease assessment scale is, because you have different people doing these ratings. I assume you didn't drive to Canada every time.
No. So this rating scale is previously established by SIUC, so Southern Illinois University, and it's a 1 to 9 scale, with a 1 being it's based on chlorosis, necrosis, and then when it gets later, it gets into percent leaf defoliation. So the 1 is from 1 to 10% chlorosis, and all the way up to the 9 scale, which is complete premature defoliation.
So he's asking is, are you sure that Dean was as accurate as you were on your ratings? Like, how did we ensure that we had?
We all use the same scale, and we hope. I mean, they are also they work on SDS as well. Those labs do. So hopefully, I don't know.
It's a pretty established scale, Mark.
Yeah.
But
It's in the literature.
and we do have pictures, just like the root route one. We have pictures, and you know, people share things, but I don't think we necessarily went out of the way to make sure that it was accurate. So.
But there's a sheet that has photos for each category, so everybody usually refers to that.
Visual
like
scale,
visual
that's kind of
scale.
where.
Yeah.
Oh, okay. Yes.
And everybody had that when they were in the field.
Most of the people outside of SDS.
All these things on the chart.
What about my friend soybean cyst nematode? Did any of the sites see anything?
So we actually did check that, and so because different locations have different ranges of what they consider low, mid, and high, and from what I remember, every location that we had, we got lucky, and they were all considered in that lowest category. So that, yeah. Chelsea.
The high and low rates of Fusarium that you use as the SDS and SCN rate.
Correct.
And
Correct.
SCN's a low rate, right?
Yes.
So since you don't see
I would personally say that this isn't, we would need more data to back that up across more locations.
This is follow-up to the
Mhm.
question. What would you say to a farmer if you were presenting this data and it showed that one field or one year where the fluopyram had worse than the base, and you had fluopyram and you had more yield loss? What would you say to the farmer in that case? If the farmer asked a question about, should I not use fluopyram because it's making my problem worse?
So I guess after hearing all the extension talks from Darren, how I would go with that, I would still say that it still is a calculated risk. The fluopyram does cost more, but if your field has a high level of SDS, it has a history of high SDS, along with that the year, as the weather plays a huge part of it, is conducive to SDS, I would still recommend it. What would you say to that, Darren? It was one location. like that's the thing. I know I only showed 4 locations, but yeah.
Yeah, he's learned too much. I mean, yeah, we just go back to the averages, Bart. I mean, there's going to be exceptions with every product every year. Overall, I think we usually see 80% of the locations have a reduction, and about 10% have it pretty break even. But you're still going to end up. It's biology, it's science. So
I kind of dispute your idea about the weather prevailing at the site though, because this is a seed treatment, right?
Mm-hmm.
You can't really see what the weather's going to be when you're deciding on seed treatment, can
No, you can look at you can look at climate, but you know how weathermen are. They can only be as accurate as what's going on right now.
most of the decisions for seed treatments are made this month or the next couple months. So you have no idea what it's going to be.
So the fluopyram, do you see bumps of resistance, or do you think of exactly reason?
So we did not look at that in our study, but from everything I've read, this fluopyram is actually a succinate dehydrogenase inhibitor. And something about the olivoflupyram is different from the other flupyrams on the market. And the other flupyrams on the market have shown some resistance, but this one does not. That's kind of out of my realm, but I have read that paper about that.
My question was basically on the seed treatment. What was your standard basic seed treatment with the fluopyram? First is high and low. What is exactly your application rate?
Oh, that was what Chelsea was referring to. So the high rate of fluopyram is actually labeled for SDS, while the low rate of fluopyram is only labeled against SCN. So it's like 0.15 milligrams AI per seed for the high, and the point is half that. So 0.075 milligrams AI per seed for the low rate of fluopyram. Jared.
Do you notice a yield drag? Is there any sort of yield differences among those resistant varieties? Has that played into
I mean, each different variety will have a different maximum theoretical yield. Is that what you're referring to?
Well, so like, does a resistant variety have like a significantly lower amount of, I guess, like, if you have low selection pressure or disease pressure, how's the yield?
I'm not familiar with that sort of data. Apparently Darren is not either.
It's not known.
Mohsen had one earlier?
Yeah, my question is about yield losses.
Mhm.
So you figure out like there are 3 different factors which are most affecting the yield.
Mhm.
How did you figure out those?
So we figured that out using the regression. So we actually use a stepwise linear regression, which actually analyzes all of those different variables that we collected. And it only uses the ones that best fit the model. So those three explanatory variables were the 3 that popped out.
So stepwise regression analysis has a problem like it cannot like address the multicollinearity issue. Did you check that? Multicollinearity.
No, I did not. What is multicollinearity again?
Like if all those variables, they are highly correlated with each other.
Mhm.
And when they are highly correlated with each other, they don't fit the model.
Mhm.
So might be they exclude some variable which might best explain, but due to their collinearity with each other, they might exclude that.
Yeah, it gets overshadowed by the other.
Yeah, so that's just like.
Yeah, no, we did not, but thank you for bringing that up. Anybody else?
Will you contribute your differences in your root rot percentage reduction between the greenhouse and the field trials?
So the greenhouse plants were only growing for 30 days. They're only like this big. Where the root rot was collected at the different timings out in the field. And remember, the most important one was at R5. So we're talking plants, big plants that are already at R5 versus plants that are only 30 days old. So I guess that would be a big explainer of the difference, of the variability in root rot.
So kind of back up to the greenhouse experiments, one of them at least we had like 70–80% root rot, and we didn't report any foliar symptoms. So I'm just curious, what did those plants look like? Did they above ground?
They were.
Did they look healthy? Did they look
They did. That was the frustrating part. But once again, we set the water bath at 17 to 19 degrees Celsius, which is more conducive for the root rot phase than for the foliar phase. But yes, they were completely healthy. I know Sharon even took a look at them too, and there was no foliar disease.
Are you willing to say, based on your field experience and other experiences, that people who are trying to breed for SDS resistance are going about it the wrong way? I mean, would you be that bold to say that in the discussion section? Because you said they're rating based on foliar symptoms, and that there's an implication in your data that might be the wrong way to go.
So I know enough plant breeders to where I would never personally say anything that strongly, but I would highly recommend looking at the root rot phase as well. And there actually is a few students that are looking at the root rot phase. In Danny Singh's lab, there's one I know of in particular he is on my side of trying to push, like, hey, we should also be looking at the roots. But it is much, much more time consuming.
For fields that you use that are naturally infested, is there a way to determine the infestation or the
yes. So
the infection level throughout the field to see if it's uniform?
So for the nature of the beast, as it is soil-borne, uniformity is not something we can guarantee. However, we did pick locations especially for the naturally infested. We picked locations with a history of SDS, and that's pretty standard for picking the different field locations. You go someplace with a history of
history of SDS. Is it like this corner, or like do you look at like previous seasons?
We look at
abundant was it throughout the field?
So we look at the particular location. I don't personally do that for picking out the locations, but to check for the uniformity, that was the picture I showed of Roland, Iowa, with the check strips, so that can help show uniformity. And that's also why we have to block everything out in the field. Because there was, on that picture, I know it's not as good on this projector, but there is a spot where it's very high SDS and like an entire block's worth where it wasn't as bad, but still there, and then it was very high again. Because it was a hill on that field.
So Jerry, the way those experiments were set up, between every set of the reps were set was for susceptible. So and the same one, so that we can compare left and right of those plots, so like how much SDS, not quantitatively, but we see it.
Were your muscatine experiments in farmers' fields or at the
So it was at the research farm, so the soils there were much sandier. And that's what we were going for at those locations.
And did you say early on that you artificially infested those as well because it was university land? Were you able to do that?
Stith was muscatine. Muscatine was artificially infested as well.
Any final questions?
Well, why did you pick such an oddball soil? I mean, muscatine is not representative of most of the soybean growing areas in the Midwest. It's sand and gravel.
I don't know why that particular location was chosen. I apologize.
Irrigation mark, so we can water it. And where's that something?
Oh yeah.
Darren likes to get melons.
Right. Other advantages from muscatine.
We brought back the
drive.
Yes.
All right. No final questions. Let's get down to the